Hi, I'm Dr. Brian Kaufman, a retired family doctor and a CLL patient myself, and the co-founder, executive vice president, and chief medical officer of the CLL Society here at ASH 2024 in San Diego.
And I am Valentin Ortiz-Maldonado, also a doctor. I'm working on CARTT cell therapy in Hospital Clinic of Barcelona, Spain.
CAR-T therapy has been incredibly exciting, and there was some fabulous successes. And I was just at one of the ASH oral presentations where people from UPenn presented patients that are alive 10 years later having CAR-T. But this is still the minority of patients for a lot of reasons. A lot of patients don't do as well with it in CLL as compared to other blood cancers. But you're working on a different process, a different kind of way of making the CAR-T that may, I kind of say, democratize it, get it out there and make it quicker. So tell us a little about what you see as the challenges of CAR-T and how your research in this sort of new way of doing CAR-T might be different.
Sure. Well, so as you know, most patients with CLL will eventually end up relapsing at some time of their life when they do not respond to targeted therapy. So CAR-T cell therapy, it was curious because it was one of the first indications that received CAR-T cell therapy, but most of the patients end up relapsing. So one of the reasons why people believe that CAR-T cell therapy may not have been achieving comparable responses to other B-cell malignancies is because it depends on the fitness of the T cells of the patient. So the drug is the own patient's T cells, which are modified to target the disease.
They're genetically re-engineered in the traditional way is they're taken out of the patient and they're sent to a central lab. And
exactly. So that usually is a very complex procedure. So you need to take out the own patient's T cells, which are cells of the immune system that are naturally having a very important paper in protecting yourself from external enemies, let's say virus, but also internal enemies, let's say cancer. So what we do is we take those T cells outside, those are usually non-malignant T cells, and we introduce a new gene that reprograms the efficacy, the specificity of these T cells, and now they are able to recognize specifically CLL cells and target them. So what we do is we try to improve the fitness of these T cells. So CLL patients do have like a reduced fitness of their own T cells.
I've heard them called exhausted T cells, that they have an exhausted profile. They're just not as good at recognizing threats and killing the threats off.
Exactly. They mention it as exhausted. They can also mention it as dysfunctional. That's why CLL patients tend to have also many other cancers and many other infections. So what we are doing is we are taking the T cells from the patients in the same way that other CAR cell manufacturers, but we are manufacturing, modifying these T cells in our very own hospital. So we're not sending or shipping the cells very far away, which also requires to freeze the cells. T cells don't like that. It's usually another thing that hurts T cells' fitness. So without freezing the T cells, we manufacture this CAR-T cell product in a very, very short time with only 7 days. So what happens is by doing this, you are able to produce your drug without freezing the cells. You can give them back to the patient without ever having to freeze them. So they are fresh, they are fitter, and what we are able to see is that the fitness of the T cells from the starting point when you collect them to the point when you give it to the patients, they improve the proportion of early phenotypes, which is usually...
Remind it, what phenotype, what are you talking, what does that mean?
When we mean early phenotypes, we mean like younger T cells, fitter T cells, T cells that can potentially lead to longer persistence and better efficacy. So by doing this, we're able to treat all our patients in just 7 days.
And sometimes, not in CLL, generally it's a slow-moving or indolent cancer.
Exactly.
But sometimes in Richter's transformation, the difference between 7 days and 3 or 4 weeks could be the difference between life and death.
Totally. So most of the patients, the ability to receive this therapy will not depend on only receiving it within 7 days. But then you might have a 10% of patients that will do. And there might be other patients that will be able to receive the CAR T cells in, let's say, 1 month, but the disease will progress. So you will always want to get patients to receive CAR T cells as soon as possible. It doesn't matter how good they are, because time can only lead them usually to more disease, and we try to avoid that. So especially for Richter transformation, who is a more aggressive disease, it is really important.
So do you have some early results that you'll be presenting here at ASH that you can share with us?
Sure. So we have enrolled our first 15 patients that have received this type of therapy. So we have treated 6 patients with CLL and 9 patients with Richter transformation. These patients were, of course, patients with a lot of disease, very refractory. We are meaning about a median of 4 prior treatment lines for CLL.
Very tough patients to treat.
Very tough patients, and a median of 3 prior treatment lines for patients with Richter transformation. That is very, very high risk patients that usually don't get a chance to get a cure at that point of the disease. And also, all of these patients, all the CLL patients, have been relapsed or refractory to both BTKIs and BCL2 inhibitors. So they really are composed of patients with really, really bad prognosis.
Right, so ibrutinib, acalabrutinib, zanubrutinib, pirtobrutinib wasn't working, and venetoclax wasn't working also.
Exactly, so that was tried, we didn't work, and then they got CAR T cells. So what we saw is, first, we were able to manufacture CAR T cells to all these patients.
Which isn't a given, right?
Exactly, so it's more likely in this type of disease of getting what we call manufacturing failure, so that you try but you're not able to manufacture the drug. But in this group of patients, we were able to manufacture the drug to all the patients. We were able to give the CAR T cell therapy in a median of 7 days. That means collecting T cells from the patient, and the very next day, we start with a lymphodepleting chemotherapy.
And that's where you give chemotherapy so the body won't reject the CAR Ts coming back into them that have been genetically changed.
So it's like chemotherapy, which is not precisely for the CLL treatment. It can also work on CLL, but it's not given for that reason. Right. It's given to allow the CAR T cells to engraft, to have more room for them to fill, to grow very high inside your body, and to really expand in the long run, and avoid rejection as well. So after that, we collect T cells, we start lymphodepletion, we give 2 days of a washout period of this chemo, and then right away we take the cells from the system where we manufacture, and we give it freshly without requiring to cryopreserve these cells, and we give them in a fresh manner in a single infusion to the patients.
Wow. So do you have some results on response rates?
Yeah, so response rates are so far, we have that more than 80% of patients do respond. And we are talking about both CLL and Richter transformation.
Wow, that's impressive.
Yeah, so it's more than 80% response rate with 67% of complete responses.
Wow, so 2 out of 3 complete responses.
Yeah.
Wow.
And most importantly is that remissions seem to be durable. So to the last time that we made a data cutoff and analysis, 80% of patients that responded remained in response. So far responses seem to be durable.
And what is that time frame? What was the average duration of response that you have at this point? Because I know it's early in the research.
It's early in the research. The patient that we have with longer follow-up is 15 months. So it's still a little bit early on, but still they're very encouraging results for this moment and for this particular type of population.
Yeah. And is there the persistence of the CAR T cells? Do we know that? Because that seems to be pretty related from what I'm learning at ASH. They need to stay around and play whack-a-mole with the cancer. Yeah.
Exactly. So usually we like to see CAR T cells persist as long as possible. So again, almost all patients have engraftment and long-term engraftment. So we have been seeing CAR T cells detectable even in the last follow-up. So at least 15 months of follow-up. Again, this is very encouraging results, but we will need more time to see how long they last. But really what we want to see is the duration of response, because we can also see patients losing CAR T cells, but not necessarily having relapse.
Now, CAR T is not a benign therapy, so we've seen what's called cytokine release syndrome and neurological toxicities. Tell us about the adverse events you've had. Have you lost any patients in this trial?
Yeah. So, as you mentioned, CRS and neurotoxicity are two class effect toxicity of this type of therapy. It's basically an inflammation. So when you give CAR T cells, immune cells from your body, and they get inside your body, they recognize all this disease, they react towards it. So this makes these CAR T cells to proliferate, reproduce, and to attack these CAR T the disease. When this happens, that makes your body get inflammation. It's like when you have flu, you get fevers. That fevers is not provoked by the virus itself, but for the reaction of your immune system to attack the virus. So in a very similar way that happens with CAR T cell therapy. So we see CRS, which is high fevers and maybe also low blood pressure. But importantly, in this type of therapy, we have only seen fevers. So we have not seen any severe degree of this complication, which is very important, especially in this population, because the median age of these patients tend to be more higher than other types of diseases that receive CAR T cells. We're talking about 68 years, around 70 years of age.
What about the neurotoxicity? Has that been an issue?
And then the neurotoxicity is maybe the most interesting part. So neurotoxicity is very frequent with several types of CAR T cell constructs. But it has been a surprise for us, and we really don't know why we haven't seen any degree of neurotoxicity.
Wow.
Not the most slightly way of neurotoxicity. So it might have something to do with the freshness of the CAR T cells. It might have something to do with the early phenotype of this type of product, but we don't really know. The good thing is we don't see any neurotoxicity.
And other adverse events, has there been any infections seem to be an issue because it's very immunosuppressive and there's been some cardiac concerns?
Exactly. So we have seen patients with infection. So infection is really one of the most important toxicities for CAR T cells. So it's very usual that people focus on neurotoxicity and CRS because there are toxicities that people are usually not used to. But infections are really, really frequent. So we have patients with infections. In fact, one of the patients that the reason of death was an infection. So this is something that we tend to see in CAR T cell therapy. Around 2/3 of patients that die because of the therapy is because of infection. Of course, we don't really know if this is only because of CAR T cell therapy, but also on the disease itself has a predisposition for infections. So it's really a complication that can occur. Unfortunately, it's not that frequent, at least in these 15 patients, but it's definitely something that has to be taken care of.
Are there trials open now? This is pretty exciting for patients if they wanted to find out more.
Well, this trial, so it's a phase 1 trial, so that means that the objective of this trial is to find the maximum tolerated dose, which has been selected. And once you finish this very relatively small trial, then you need the patients to have months and months to see how they develop. And when the data is mature enough, you can go to the regulators and ask for the start, if they think that the outcomes are met, to ask for a start to a phase 2 trial. So the next step will be to start a multi-center, multinational trial. And that will be probably, if this happens, will eventually lead to, hopefully, some time of approval.
Wonderful. Any final thoughts or messages that you want to give to CLL patients or Richter's patients about this exciting new way to do CAR-Ts?
Yes, so the message will be that we are still learning a lot, we are improving a lot, and we can see how every year we see something new. So with these small improvements, we see how we are improving again and again a little bit the prognosis of CLL patients and Richter transformation patients. I do believe that even this type of generation will be able to achieve cure or long-term remissions in a significant proportion of patients. But time will show this, and hopefully this happens soon.
Thank you so much for the research you and your colleagues are doing. This is very exciting. Thank you so much.
Thank you for having me.